Hello future scientists, welcome to Cosmic Edutech by Dr. Priyanka, and you are watching this series on stainings in microbiology. So today I am here with yet another staining technique, that is acid fast staining, also known as Ziehl-Neelsen method. And today we will learn the principle, procedures, observations, applications and some easy tricks to remember this method. Now, this technique is very much crucial, especially in medical microbiology, in detecting mycobacterium, the causative agent of tuberculosis and leprosy. This acid fast staining is a differential technique used in medical microbiology. Now, this particular method is going to divide the bacteria into two groups, that is acid fast and non-acid fast. So, it is particularly important in clinical microbiology, especially for identification of mycobacterium species and certain parasites. Let's try to dive into the principle and understand the working behind it. Now we are specially studying mycobacterium using this technique. So this mycobacterium, it has a it has a cell wall that is rich in something called as the mycolic acid. That's a waxy lipid, and it does not allow these dyes to easily penetrate inside. So here we have to use certain specialized stains like carbol fusion. Now this carbol fusion, it penetrates inside or it enters inside the bacterial cell wall when heat or phenol is applied. And when it is treated with acid alcohol, the non-acid fast bacteria, they lose the red color of carbol fusion. And when we are using the counter stain like methylene blue, they will take up the color of methylene blue. And hence this non-acid fast bacteria, they appear blue in color. So how do we remember it? So I'll give you a simple trick, that is red mycos are rebels whereas blue are obedient. So red myco rebels, meaning the acid fast bacteria, they will take up the red color because of heat or phenol, and they will not let it go out even when acid alcohol is being applied. So, if the color is lost, so what is remaining? The counter stain. If it is non-acid fast, they are going to obediently take the blue color. So, that's why blue obeys. Let's see what are the materials that we require. Materials, just like any other staining method, are very easy. The primary stain, the counter stain, a decolorizer, and obviously slides and a very good quality microscope. So we will require a bacterial smear, then carbol fusion, that is the primary stain, a decolorizer that we are using is acid alcohol, counter stain is methylene blue, and a microscope. So now let's see what is the procedure. Now initially I have used the name that is acid fast staining, but this method was originally developed by a person called as Ziehl, and later on it was modified by Neelsen. So that's why collectively the name comes, that is Ziehl-Neelsen method. So Neelsen used carbol fusion from Ziehl's experiment, and with added heat and decolorizing agent like acid alcohol, and then he used methylene blue as the counter stain, and he developed this technique. So together collectively it is called as the Ziehl-Neelsen method. So coming on to the next part, that is why it is acid fast. Now here we are using acid alcohol as the decolorizer. So because of that, the technique has earned the name, that is acid fast technique. And because heat is being applied, it is also called as the hot method of acid fast staining. So it is synonymous with the name Ziehl-Neelsen staining. Let's go step by step through the Ziehl-Neelsen hot method. So first of all, we have to prepare a smear and heat fix it. Now mind you, the smear should be a thin one because we do not want any sort of overcrowding over here. Next, flood the slide with carbol fusion and heat it gently until steaming. Okay, but you should not allow it to overboil also. And after that, we have to perform this for almost for like around 5 minutes. After that is done, allow it to cool and rinse it with water so that the excess stain around is washed off. Then the fourth step is adding a decolorizer. So decolorizer that we are going to use here is acid alcohol. and we keep it in contact for around 15 to 20 seconds, and later on we will wash it again very gently so that the excess that is being oozed out is washed off. Then finally the sixth step, that is adding the counter stain, that is methylene blue. We keep it in contact with for around 1 minute. So after this it is being nicely gently washed, blown dry and blotted dry, and observe it under oil immersion microscope. We are supposed to see a colored combination of pink and blue. Suppose it's a mixed culture, because we know acid fast bacteria, they are going to appear pink because they want to retain the carbonyl fusion, whereas the non-acid fast bacteria, the carbonyl fusion will be oozed out by using the acid alcohol, and they will take up the counter stain, that is methylene blue. So Ziehl-Neelsen method, it's a hot preparation or hot method. There is yet another variation of this method that is called as the Kinyoun method or the cold method. So here everything remains the same. Only thing, we are not going to use any sort of heat over here. So this method, it avoids using heat, and rather it uses stronger concentrations of carbonyl fusion and phenol that is going to allow it to enter inside the waxy mycolic acid, and also the time for which it is kept is little longer as compared to the Ziehl-Neelsen method. So this method, because we are not going to use any sort of heat, it becomes very safe for, especially for labs, because your open flames are not recommended. So as we have already discussed how this stain bacteria would look like under the microscope. So under the microscope at around 100x magnification, acid fast bacteria, they will appear bright red or pink rods. Whereas the non-acid fast bacteria, they appear blue against the background. So obviously we know the reason and principle why the difference in coloration occurs. So let's see some of the do's and the don'ts here. It is very important, as I have already mentioned, that we have to use a thin smear, because it will allow better penetration and you will get very good clarity in the result. Next is heat gently. It should be approaching steaming but not boiling, right? And finally, you always use a control slide. The reason why we need to use a control slide is it helps us in differentiating the colors. Sometimes it causes confusion. So it is always advisable to use a control slide. Let's see what are the don'ts. So do not overuse the decolorizer, because it can give you false negative results. Then always be firm and careful while washing. So because otherwise carryover stains may interfere. Then next is never overheat the smear, because it can destroy the cell and you may not get the expected results. Let's see some of the applications of this Ziehl-Neelsen method or the hot method of acid fast method. So it is specially used in the clinical microbiology for diagnosis of tuberculosis, leprosy, okay, or Nocardiosis. Then apart from this, it is also used to detect the acid fast oocytes, especially in the stool samples, and thus this technique is very much valuable for the medical microbiology. Move on to the conclusion, let's try to understand, or let's try to get revision of certain Viva questions that can be asked for your practical exams. So like I'm giving you some of the expected questions, like basically you need to know the principle of acid fast. Then why do we apply heat in Ziehl-Neelsen method, and why it is different from the cold method or the Kinyoun method. So then there is, you have to remember some of the diseases and the causative agents which can be detected using your acid fast staining. And then the color of the acid fast versus the color of the non-acid fast, because the examiner can ask you in a twisted manner. Then again, why gram staining is not used? It is because of the mycolic acid. So these are some of the things that you need to be prepared, because it makes you more confident to answer your Viva and also the performance in practical exam. So let's see the conclusion. So in conclusion, acid-fast staining is a powerful diagnostic technique, and always remember the trick, that is the acid-fast, that is the red rebels, and non-acid-fast are the blue obedient. Good boys. Okay, so this simple line will help you to recall and easily remember it. So thank you for learning with Cosmic Edutech. Do not forget to subscribe for more such biology and microbiology hack and exam smart tricks. So now all the best for your practical exams, and also at least the 5-mark questions in your theory exams will be completely covered with this lecture. So till then, take care and keep smiling.